I think we can go ahead and get started. So hello everybody and welcome to Scripps Research, fully interactive resources for students and teachers or FIRST. My name is Rosie Alvaran-Seckler and I will be your moderator this afternoon. For those who are joining us for the first time, Scripps Research is one of the largest nonprofit private biomedical research institutes in the nation, and our mission is to develop the next generation of medicines and diagnostics to address the most present health problems in the world. For those who are joining again, welcome back. The topic in today's webinar is Careers in Biomedical Research, in which our speaker will be highlighting the many roles a scientist with or without a PhD can have in a research institution such as ours. Before we begin, we would like to remind everyone about a few housekeeping items. All participants have been muted and your videos have been disabled. The format today will consist of a 15-minute presentation followed by a 10 to 15-minute Q&A session where we'll be taking your questions live and we will be answering them live. To do this in the Zoom platform, you would like to take a look at the bottom of your screen. There's a Q&A button where you can type in your questions. You can do this at any time and you can do this anonymously. We will be answering them though on the second part of the presentation. Today's speaker is Dr. Joanne Doherty. She's a staff scientist on our Florida campus in the laboratory of Dr. Joseph Kissel in the Department of Molecular Medicine. The overarching goal of her research is to understand the molecular mechanisms of cancer development and growth in order to identify rational drug targets. She received a PhD from the University of Maryland for thesis work on DNA tumor viruses and completed postdoctoral training at St. Jude Children's Research Hospital for developing genetic models in African clawed frogs. Welcome, Joanne.
Thank you, Rosie. I'm going to share my screen now. Thank you for that introduction, Rosie. Hi, everyone out there. My name is Joanne, and today I've broken up this talk into two sections. The first part, I'd like to talk about careers in science and how I got to where I got to, and maybe give you a little bit more information about what's available. So that's more of a career talk. And then the second part of my talk, I'll tell you a little bit about the research that I'm doing in Joe Kissel's laboratory. All right, so a lot of people who are interested in science want to know what the difference between academic versus industry research. And at a place like Scripps, we do research that is very similar to industry or pharmaceutical research. And that's what we call applied research. So it's the development of drugs or vaccines or diagnostic tests, anything that can then be used to treat human disease or health. And a lot of that type of research is also done in an academic setting. So the main difference between academic and industry research would be where the funding comes from to do that research. So in industry, the funding comes from investors or the profit from the sale of their products. So in an industry setting, their research has to be very focused on doing research that will eventually result in a product that can make money. And if they don't, then the business goes out of business. So they do have to be very focused. The funding in academic research, on the other hand, comes from mostly national funding. So the National Institute of Health or the National Science Foundation, they provide most of the academic research funding in the US. There are also private funding agencies. As an example, I'll put the Gates Foundation. So these are much smaller funding agencies, and they generally target specific diseases. But because of this funding source, academic research isn't restricted to generating a profit. Sometimes it does, but they're not limited to generating something that's going to make money. And because of that, the research can be more expansive. And the research can go after basic questions in biology and chemistry, just for the sake of understanding how these systems work. And that eventually can lead to applied research. So there are academic laboratories that do develop drugs, and they then go on to start their own company and become an industry laboratory. But in academic research, there's also the opportunity to focus on orphan or rare diseases. So these are diseases that affect few people in the United States and the world. Because of that, industry can't go after those diseases because they're not going to get a return on that investment because there won't be enough people buying their products to make up for the research money that they spent. But of course, the National Institute of Health is very concerned with all human health, so they do fund a lot of research into rare diseases. And the disease that I work on now in Joe Kissel's lab is considered a rare disease. And because of the national funding, we're able to understand more about it and hopefully help people with that disease. Okay, and I put this arrow in there to show that there is a lot of similarity and there's a lot of give and take between academic and industry research. This is a typical career path for someone who's interested in doing academic research. So you'll start at a high school level, you will go to college, get some level of college education. If you're interested in running your own research laboratory, you'll need a bachelor's degree, either a Bachelor of Science or a Bachelor of Arts with maybe a minor in science. From your undergrad, if you're focused on this, you'll go on to graduate school. Graduate school is anywhere between 3 and 6 years of education. And that involves a minor amount of time on classwork and a major amount of time on research work. So you'll spend most of your time in graduate school working on a research project in a laboratory. If you're interested in going further into an academic faculty position, you'll need to do a postdoctoral training. And what that is developing your own independent research project under the guidance of a mentor. So it's guided independent research, and that prepares you to run your own academic research laboratory. At the faculty level, you're then required to generate enough money to run your own research program. And you do that by writing grants and papers, either through NIH or those private funding organizations. But of course, not everyone goes through to become faculty. At each level, there are opportunities for people to do academic research in a laboratory. So at the bachelor's degree, you can go into a laboratory if you have a strong background in science. You can participate in laboratory research. There's also associate in science degrees, which are 1 to 2 years focused on a specific area of science. And you can also do that after high school and work in a laboratory. After graduate school, you have much more exposure to research so that you can then use those skills to get into other different levels of research. And what I did was, after my postdoc, I went into research without becoming a faculty. So although this is the traditional pathway to faculty, there are lots of opportunities throughout this process. So my path started with an undergraduate degree in microbiology and immunology. And my summers I worked in a clinical microbiology lab. So the clinical lab was very regimented and standard. I did not like that. The other thing is, as an undergraduate, I did not get a lot of research experience. So I wasn't really sure if I liked it or if I would like it or not. And I think what you're doing today, learning more about it through some of these seminars, is really important to do at the high school level, so that when you go into college, you can start to look for some of these opportunities to do internships and research outside of your classes. And I think that's an important thing if you think you're interested in research, to try and look for those opportunities. Unfortunately, I didn't have them. So when I graduated, I took a job as a research technician in a laboratory. And this was a cell biology laboratory, and it's really where I decided I did love to do research. I loved the entire process, from reading the literature, understanding what we know, what we don't know, and then designing experiments to answer the questions that we don't yet understand. So that's what really excited me about research when I took that job. I wanted to get more involved in it, so I got a PhD in molecular and cell biology at the University of Maryland. And at this stage, I studied cancer biology. And I liked that, but when I finished my PhD, I felt like my expertise was too focused. So I did a postdoc in a different field, which was developmental biology. And this was actually very helpful because it helped me put cancer biology in perspective of developmental biology. And there is a lot of there is a lot of crosstalk between development of cancer and basic embryonic development of humans. So that did help me. But at the postdoc level, I realized I did not want to write grants and become a head of a laboratory. So when an opportunity came up to become a staff scientist here at Scripps, I jumped at that opportunity. And since I've been at Scripps, I've worked at several laboratories related to cancer biology. But today, I'm just going to tell you about the lab that I'm in now, where we study a tumor suppressor that results in a schwannoma, which is a tumor of Schwann cells. So that's what I have for my career aspect of being a scientist. One more slide. My role here, I would say my role is to advance the research projects in the laboratory. And I do that by designing and running experiments, developing techniques and reagents, writing papers, and helping to write grants. So really, I'm helping Joe move his research program forward. The other main role that I have is to train and mentor personnel in the lab. So before COVID-19, we had interns, high school interns, college interns. We also have graduate students, and we don't yet, but eventually we will get some postdocs. So a big part of my role is to help those people when they need it. And that's it. That's it for my role. What I'd like to do now is to switch and tell you a little bit about the research that I do. So in Joe Kissel's lab, we study neurofibromatosis type 2. This is a genetic disease caused by mutation in this gene NF2. It's a tumor suppressor, so when it's mutated or lost, it results in the development of a schwannoma, which is a tumor of the Schwann cells. Schwann cells are these cells that wrap around neurons, and what they do is protect the neuron and insulate it so that electrical currents can pass through the neuron. Patients with this disorder, they develop schwannomas on the cranial nerves, which are found not in the central nervous system, but in the head region. And the acranial nerve, where these tumors predominantly develop, is responsible for hearing and balance. So when the tumors form on the cranial nerve, the patients lose hearing and lose their balance. You have peripheral nerves throughout the body, so patients will also develop schwannomas in some of these other peripheral locations, as well as along the spinal column. So this is a MRI showing the cranial schwannomas, and it's bilateral cranial schwannomas. You can see here, this patient also has a schwannoma on the sciatic nerve. And you can see small schwannomas developing on the dorsal ridge ganglia. So the treatment for these types of tumors is surgery. And you can imagine doing surgery in this cranial location from a tumor that's wrapped around a nerve, you're going to create some nerve damage. So the surgery to treat, to remove the tumor, generally, or in many cases, results in hearing loss as well as balance disturbances. So in the Kissel lab and in other labs, we're interested in trying to find therapeutics to treat these tumors so the patients don't need to undergo surgery. And some of the tools that we use in the Kissel laboratory, experimental models would be cells in culture, as well as animal models, genetically engineered mice, or mice that we do tumor transplants into. With these two models, we can knock out genes with CRISPR or siRNA, or use the genetically engineered mice, or we can do gene overexpression through transfection or the transgenic mice. And with the models, we can then look at proliferation or tumor growth, gene and protein expression, or the sensitivity to known drugs. What we use these experimental models for, we then want to confirm in the human tumor. So we want to make sure that what we've tested in the models is possibly also occurring in human tumors. And with the human tumors, we can look at gene expression or protein expression. So with those models, we came up with a hypothesis based on what was known about how these tumors develop. So NF2 is the gene. The protein for this gene is called merlin. We knew that merlin regulates a transcription factor called YAP, which binds to TEAD. And YAP and TEAD regulate the expression of growth and anti-apoptotic genes. So NF2 merlin inhibits this and then inhibits the expression of growth and apoptotic genes. In the patients with the disorder, this gene and protein are lost, enabling YAP and TEAD to upregulate the expression of these genes and cause tumorigenic growth of those Schwann cells. Recently, another group has identified this bromodomain protein, BRD4, is recruited to genes by YAP. So BRD4 binds to YAP, and YAP brings it to the promoter region of genes. BRD4 contains these bromodomains that bind to acetylated histones. So we think that YAP is acting as a transcription factor through recruitment of BRD4, which then recruits basic transcription factors, TFP. So about 10 years ago, an academic group identified this small molecule called JQ1. And JQ1 is a very good inhibitor of bromodomains. So it can inhibit BRD4 by binding to the bromodomain and blocking it from binding acetylated histones. So we wanted to know if using this inhibitor JQ1 or inhibitors that block bromodomain proteins could be an effective therapy for schwannomas. So we took human schwannoma cells in culture and we treated them with JQ1, and you can see if we measure the amount of cells over time, the proliferation is inhibited when we treat with JQ1, whereas the control treated continue to grow. We also did knockdown studies because there are several bromodomain proteins in the family. And JQ1 will inhibit all three of these, from domain 2, 3, and 4. So if we knock down BRD2, BRD3, or BRD4 and look at proliferation, we see that only knockdown of BRD4 causes inhibition of proliferation. So that suggests to us that JQ1 is inhibiting the Schwann cell proliferation by inhibiting specifically BRD4. We also looked at the growth of schwannoma cells in animals by doing transplant experiments. So we transplanted a luminescent engineered schwannoma into mice in the flank. And we treated them with vehicle or JQ1. You can see the tumors grew in the vehicle-treated animals, shown by this luminescent signal. And the signal is decreased in the JQ1, indicating that the schwannoma cells were not able to proliferate when the animals were given JQ1. At the end, the tumors were excised from the animals and weighed, and you can see the JQ1 treated tumors are a much smaller size. So then we wanted to find out if BRD4 really was playing a role in human schwannomas. And we have a collaborator that provided human schwannoma tissue that we were then able to do immunohistochemistry for BRD4. And you can see the brown staining is the BRD4. This is median levels of BRD4 staining. This is high levels of BRD4 staining only in the schwannoma tissue. When we compare it to a normal nerve tissue, we see very little BRD4 expressed. So we believe that targeting BRD4 with JQ1 or a bromodomain inhibitor might be effective at treating human vestibular schwannomas. In the human vestibular schwannomas, they have lost the NF2 tumor suppressor. So we took our Schwann cells and used CRISPR to delete the NF2 tumor suppressor. And we then treated these two different cell lines with JQ1 and looked at cell proliferation with increasing concentrations of JQ1. And this is what we call a dose response curve. And from this, we can calculate the IC50, which is the concentration of JQ1 with we have 50% reduction in proliferation. And you can see the NF2 null cells, that dose response curve occurs at a lower concentration, it goes down at a lower concentration of JQ1. So the IC50 for the NF2 null cells is lower, indicating that the NF2 null cells are more sensitive to inhibition with JQ1. This is what we would hope to find when we treat patients, because not all of the patient cells are NF2 null, only their tumor cells are NF2 null. And if we can have some specificity for targeting the tumors over normal tissue, that would be ideal. But we wanted to know more about what was causing this sensitivity, so we did a RNA sequencing experiment. We took our wild type cells or our NF2 null cells and treated them, either control treated them or treated them with JQ1, and then we measured all of the RNA in the cells. And from that, we can look at the cells that are upregulated. We can look at the genes that are upregulated or the genes that are downregulated with JQ1 treatment between the two different cell lines. And of course, you'll have genes that are common to both cell lines, but you'll have genes that are specifically down or upregulated in the NF2 null cells. And using this type of experiment, we can then start to understand what causes the sensitivity. And hopefully that will lead us to how JQ1 is functioning and potentially other genes that could be targeted in the NF2 null schwannomas. So that's a little bit about the research. And of course, research is never done as an individual. The work is funded through grant writing from Joe Kissel. Scott Troutman is a technician in the lab that has done the animal work. Benai was a postdoc who had started this project. Merlin is a graduate student who helped me with the bioinformatics. Rachel Brody provided the human schwannoma tissue, and Doug Miller is the neuropathologist that helped us with the schwannoma tissue. So that's kind of an introduction to research science from my perspective. I'd be happy to take any questions about any part of it.
Thank you so much, John, for that great presentation, for particularly highlighting the different educational degrees that students can have if they want to become scientists, and that not everybody might need to go after a PhD.
Mm-hmm.
And also for talking a little bit about your research. I know that our audience particularly likes to learn more about the kind of research that we do at Scripps Research, and also what drug discovery entails. So I see that we have two questions here. I'm going to read the first one. It's a little bit long. I have a master's in molecular biology, but have no desire to get a PhD. I've worked in academia as a research technician, and I felt that I had limited opportunities to advance my career in that setting. In industry or at research institutions, if I don't have a PhD, is it possible to advance my career beyond technician or lab manager? And what are possible positions for me? I want to thank Catherine for this question. It's a great question. So, John.
That is a very good question. And I'm going to say, when I came to Scripps as a staff scientist, I ended up working in several laboratories. And part of that is you do need to find an environment where you can advance. And sometimes that means you're going to have to look at a lot of different laboratories. So since I've been at Scripps, I've worked at 4 different laboratories. And I've enjoyed them all, but some of them I had more opportunity to advance than others. So some of the advancement, you're going to have to find the right place. I don't know a lot about industry, but I think you may have more, it might be more structured to advance in an industry setting. Because in an industry setting, they don't have the same number. They usually don't have graduate students, or some of them have postdocs, but they have fewer postdocs. So I think they might rely more on professionals than people that are studying. So in the industry, you might be able to see more advancement faster than in an academic setting. Was that the question, or was there more to it?
I think so. I think that a lot of people would agree with your answer in academia. So the second question is, what new research areas will open up in 10 years or so, if you can predict?
Well, I listened to the robotics, and I guess robotics is hot.
Yeah.
So I might have to go back to school and study robotics. Yeah, I mean, I think this pandemic has shown us that virology really is important, and I hope it stays in the forefront of funding and research, because I know sometimes once we get through these pandemics, people stop thinking about it and they stop funding it. But I do think research and studying spillover of these viruses is going to be very important in the future. That's what I have.
Great. So the next question is, in your education and career, what do you feel was the hardest obstacle to overcome?
Oh, that's a very good question.
Yeah.
I think I had the hardest time in undergraduate, and it was because I didn't have a clear idea about what I wanted to do. I love science. I love the science classes. but I didn't take advantage of independent research. I didn't find a lot of programs where I could practice or do research in another setting. And I think that was my fault. I wasn't as motivated as I should have been. And because of that, I felt like maybe I was behind. So I felt like the other people that, the other students that I knew in undergraduate, they seemed to have taken more initiative and done more of the due diligence to find out what careers they liked and what careers they could try and get more information from. And I think that was hard for me. I had to learn that. And once I finished my undergraduate and started working as a technician, that's when I was able to figure out what I wanted.
Okay. And that, I guess, a segue to that question is not here, but it's been asked before. So you kind of knew that you wanted to go to PhD. There was no doubt in that decision. It's been a question before.
Yeah, I really wanted to do that. And I think it goes back to the very first question. As a research technician, I did feel like my career prospects were a little bit limited with just the undergraduate. So that was career-wise, I wanted to do more, but science-wise, I really wanted to get deeper into the research. And I felt like to do that, I needed more education. I needed to get the PhD.
Right. So next question is, what will be your advice for someone that is interested in becoming a PI?
For someone who wants to be a PI, I think it's critical that you really develop an independent research project. And that can start in your PhD. So as a PhD, you have a thesis advisor, and in general, you work on a project that's related to their research. And that's good, that's how you learn. But at that level, you need to start thinking of your own ideas, your own questions to go after, which are distinct from your mentor's project. And if you start at the PhD level, developing your own independent ideas and programs, then when you go into your postdoc, you're going to have your own ideas about what research you want to do. And that's key to becoming a faculty. You need to have a lot of ideas. You need to be able to convince people that those ideas are going to be fruitful, and they're going to develop into results and data that people are thrilled about. But I think it has to start as early as possible, thinking about research.
Great, that's great advice. So then if we continue on the advice kind of questions, the next question is, what advice do you have for high school seniors now who are not sure what major they want to pursue, but they are interested in STEM?
So I would recommend looking at as many careers in STEM as you can. So if you're interested in biology and chemistry, then look into research, either academic or industry, it doesn't matter. If you're interested in math, look for careers in math. And try and get as much exposure. It's either hands-on exposure where you do internships, or what you're doing now is talking to people that are in that field and asking them, what do you do every day? What kind of skills do I need to get into that position? Do you like where you are? Are there opportunities for people in those jobs? It's a lot of work, but you know what you like. And if you focus on what you like and try and find a career where you can do what you like, then you'll be happy.
Great. So next question, what is the difference between working on a private research laboratory and a university research lab? I guess they mean maybe industry versus university.
Well, Scripps is a private nonprofit, and we're a little bit different from a university. So the research is at both types of institutions is funded by NIH. So the research is generally very similar. But for the faculty, it's a little bit different at Scripps compared to a university. So a faculty at Scripps has less, I would say less teaching responsibility than a faculty at a university, where they would have much more teaching, a bigger teaching load. So that's the difference between academic university research versus a private nonprofit like Scripps. But Scripps, of course, has graduate students that they train, and there is some teaching that faculty are required to do.
Great. And the last two questions, they're more about your career path. So is there a reason why you decided to work at Scripps versus industry, or like you just explained, go into like a university? I know that you have taught undergraduates and you have done that as well. So is there a particular reason, one versus another?
There isn't really. It's serendipitous. I was offered a job at Scripps, so I came here, and I love the institute. I love the research that's done there, but I also love Florida. So I've settled into this area, and there's no real reason why I didn't go into industry. Just I had an opportunity.
Great. And then the last question here, if you were to go back in time, would you still pursue this route, or you would have changed anything?
That's a tough question.
Yeah, that's a tough question.
You know, I've loved each stage. I've really loved it, and you know, it's not always great. Things are hard, but when I look back at every stage, I've enjoyed it. So I would say now that I wouldn't do anything differently, except maybe spending a little more time when I was an undergraduate looking at different careers.
Great, thank you. I think that's a great way to close. Unless you have something else that you want to add.
No, I give everyone kudos for doing this, because I think even if you're not interested, finding out you're not interested is as important as finding what you're interested in. So I think doing the looking at different careers is really important for everyone.
Great. Thank you. I want to thank you again for this great presentation, for spending time with us and answering the students' questions. I'm very appreciative of your time, of all our presenters. So thank you again. On screen here on the flyer. So the next webinar is on the role of the biostatistician, and you can register for this webinar in the same place that you registered for today's webinar. Also, you can see more content on our YouTube channel, and this is going to, we're going to try to put it in the chat right now, but it's youtube.com/ScrippsGrad. And when we close the call, you're going to be redirected to our page where you can find more contact, previous webinars, and also where to enroll to future webinars. Thank you so much, and have a good day.
Thank you, Rosie. Thank you, Nitya, Jeremy. You guys do a great job.
Thank you. Bye.